Hello students, so the needle electrode was remaining. So this needle electrodes, they are used to reduce interface impedance and as well as the movement artifacts. So basically what happens, some electroencephalographers, they used subdermal needles so that it can penetrate the scalp for EEG measurements. So they used this needle electrodes. They use this needle electrodes which can penetrate the scalp. As you can see here, it can penetrate. These are the needle electrodes. In this slide also I have shown the needle electrode. This is like this. So these needle electrodes are inserted or they are penetrated to the scalp for EEG measurements. They are not inserted into the brain. They merely penetrate the skin. They are just simply inserted through a small section of the skin just beneath the surface and parallel to it. And whereas in animal research and occasionally in man, in human beings case, what happens, these longer needles are actually inserted into the brain so that localized measurement of potentials can be obtained from a specific part of the brain. And this process, it actually requires longer needles which can be precisely located through a map or atlas of the brain. And sometimes for this a special instrument called stereotaxic instrument is used, which can hold the animal's head and guide the placement of the electrodes. And often these electrodes, they are implanted to permit repeated measurements for a longer period of time, and in this case connector is cemented to the animal's skull and the incision through which electrodes are implanted is allowed to heal. So needle electrodes, this was for EEG measurements. Now needle electrodes for EMG, it consists of fine insulated wires that placed so that their tips, which are bare, are in contact with the nerve muscle or other tissue from which measurement is made. The remainder of the wire is covered with some form of insulation so as to prevent short circuiting. Wire electrodes of copper or platinum are used for this EMG pickup from specific cells, and the wires are either surgically implanted or introduced by means of hypodermic needle that is later withdrawn, leaving the wire electrode in the place. With this type of electrode, the metal electrolyte interface takes place between the uninsulated tip of the wire and the electrolytes of the body. So this was all about needle electrode. Here I have completed the biopotential electrodes. I will be giving you the notes for biochemical electrodes. Thank you.